All right, guys. Welcome to lecture one of A&P 2. I will show you what we are going to do today. So eventually there will be overlap between lab and lecture as far as material goes, but in lab, as you'll see, we have a couple, two labs that we have to get through before we get to the nervous system. So everything that you're learning will work for both classes, whether you learn at the same time or not, which is wonderful. So today we are going to start off with the nervous system. We're going to do a basic introduction. I'm going to walk you through, try to explain kind of what's going on here. Obviously, we have several divisions of the nervous system. This is what it looks like with the entire body removed. It's very, very significant. It's responsible for sending electrical messages throughout the body for both sensory and motor information. Obviously, there'll be areas like the brain and the spinal cord where we have integration. And we'll kind of talk about what that is a little bit. And obviously, as we progress through and further lectures, we're going to dive into the function of what all these do. So basically, we break the nervous system into a few general components. The first one is called the CNS, and the second one is called the PNS. We have a sheet or a slide where I'll walk you through kind of the roadmap that we're going to call it as far as the divisions go. But the brain and spinal cord are part of the central nervous system. And I'll try to color code this for you here, brain, spinal cord, respectively, as opposed to well, that's a very bad color to use. Oh dear. I shall use a different color. The nerves, which we can see branching off here, and these are part of the peripheral nervous system. An important component and kind of a separate division itself are sensory receptors. We will talk about a plethora of them, but sensory receptors are basically receptors found in the peripheral nervous system that can detect some stimulus, whether it be, touch, pressure, temperature, things like that. So this is a pretty important division, right? As we could see here on this slide, how it's broken down. Nerves, and this is an important part. Nerves are large structures that we saw in the PNS, but they are made of the base cell unit of the nervous system called a neuron. So an important differentiation is the term nerve versus neuron. A neuron is the cell, and a nerve is a group of these cells. And I think on like the last slide or whatever, as we progress through this PowerPoint, we will see what that nerve looks like and kind of how it's composed. And in the future lecture, we'll talk about what spinal nerves are and cranial nerves and things like that. So as far as the organization goes, this is what I call the roadmap. And it's very important to understand, right? So you can see that we have a division up here for CNS, and then everything else below that is the PNS, right? So it lets me erase that. You can see that the brain and the spinal cord are found in the central nervous system. There are two things that are happening. There's constant sensory input coming into the brain, and there is constant, and I'll always try to kind of color code these for you, and there's constant motor output coming out of the central nervous system. The sensory components are referred to as afferent, and I think it will say that, yep, afferent means going to. And all messages, and we'll talk about what these are and how they're generated, all these messages going to the central nervous system will be called afferent messages, and these are all sensory. That's very, very important, right? Sensory messages go to the central nervous system. and motor messages go away from the central nervous system, right? So you can see the arrows coming in or the arrows going out. That is a very important differentiation that you must understand because we will continue to talk about it over the next couple lectures. The term efferent refers to motor control, right? These are messages, again, exiting the central nervous system, and there are two different branches. Each one kind of has its own lecture that we're going to talk about. Some of it we can control, and some of it we cannot control. So somatic nervous system refers to voluntary, you're choosing to do it. Voluntary, usually skeletal muscle control. So we can see that if we want to control a muscle or facial muscles for speaking or whatever, chewing, and your hands, obviously this all involves muscle contraction. All that choice, all that you choosing to move a muscle, whether it's during exercise or whatever, is part of the somatic nervous system, as opposed to the autonomic nervous system, which is broken into three different categories. The sympathetic and parasympathetic, we will talk about in its own lecture. These are involuntary. We cannot control them. Basically, that's going to be your fight or flight response, which is called the sympathetic, or your rest and digest, which is called the parasympathetic. And then we have a third branch called the enteric system. This is a specialized branch of the nervous system that involves the digestive system. So that's important. Obviously, the digestive system has a lot of nervous input, and we'll dive into that when we get into the digestive system way down the road. But this is basically the roadmap. So when you're studying the nervous system, it kind of helps to see, am I studying the brain and spinal cord? Am I studying nerves and sensory and motor messages coming in or out? Am I talking about sensation specifically? Am I talking about controlling muscles? Am I talking about the autonomic nervous system? So this map is important. We'll constantly come back and reference it to kind of see where we are as we are going through and studying various aspects of the nervous system. So number one, the neuron is, and we'll look at a picture of it soon, the major unit of the nervous system, clearly. But a neuron has many different helper cells. It's a lot of work to be done, right? So the helper cells are called neuroglial cells. And neuroglial cells, there are multiple different types. And we kind of talk about the function. The peripheral nervous system, the PNS, has two types of neuroglial cells that are involved in producing something called myelin, right? And we will talk about that. Satellite cells are the other type. So we have satellite cells and we have Schwann cells. You will hear me talk about Schwann cells a lot, especially as we get to this in lab. Schwann cells make myelin, and I'll show you what that does later. Basically, simply put, it speeds up an action potential, right? It's like this coat that's around the axon of the neuron, and it makes the messages go faster. But in the PNS, the Schwann cells produce myelin. There are also satellite cells that help provide support and things of that nature. In the CNS, we have four different types of or I should say neuroglial cells. The first one is called an oligodendrocyte. And their specific role, again, two branches, PNS, CNS. Their specific role is producing myelin. They do the same thing as the Schwann cells, but they're different. Number one, they can myelinate multiple axons. Number two, they're found in the central nervous system. We also have astrocytes that help with something called the blood-brain barrier. That's what BBB stands for. They provide support. Basically, what they do is they're very involved in feeding blood flow to neurons. And as we get into the brain, we'll talk about them a little bit more. We have microglial cells, which are immune cells. Obviously, they're going to go around and kill stuff. And then we have ependymal cells, which is our fourth type of neuroglial cell, found in the CNS, and they produce something called CSF. So I talked about blood-brain barrier. These ependymal cells produce cerebral spinal fluid. When we get to the brain, we'll talk about it, obviously, a little bit more. Um, but these are not neurons. They do not send action potentials. They are not involved in the electrical conduction. They're providing support. Maybe they're producing myelin in a specific location. Maybe they're fighting off some type of baddies. Maybe they're filtering blood and producing CSF. But there are a lot of them, and they're important. Now we get into the neuron itself and some physiology associated with it, right? So right off the bat, we have multiple parts of this neuron. The neuron is the cell. What this thing does is receive input, and then it decides at a specific locale that I will talk about whether or not to send a message. When this message is sent, the electrical message travels through the axon and it will come to the end. And at the end, we will release chemicals. So the majority of this is electrical, and then at the end, we'll release chemicals called neurotransmitters. And neurotransmitters are chemical released from the end of the synaptic knobs, if you will, of a neuron, and they will communicate with another neuron. And then neurons talk to neurons, which talk to neurons, which talk to neurons. And that's how the nervous system works. So a few key things that we're looking at on here are dendrites. Dendrites are the projections off the cell body. They're usually smaller and they're usually involved in receiving input from different presynaptic cells. Obviously, we're going to have a membrane. We're going to have organelles in here. One of the important organelles to mention are these little guys in here. And these are called Neisel bodies. And Neisel bodies are involved in making proteins. Obviously, the neuron is a cell. It's going to have lots of proteins made. They have these specialized structures called Neisel bodies, little patches of rough ER. And these Neisel bodies are primarily found in the cell body. And they produce proteins, which can be extremely important. Obviously, we'll talk about cytoplasm. That's the liquid component inside the cell, and the nucleus as well. So a little bit different structure than the typical cell that you're used to seeing, and primarily because of the presence of Neisel bodies and the branching aspect and how it has all these different arms, and how it's able to send an electrical message. Those are key factors, right? So this. This is a little more of a cartoony picture. So I'll show you. So maybe there are some neurons talking to this guy, right? What do? They send messages. They release these chemicals, right? And this thing's receiving all this stimulus, and this area right here, it's called the axon hillock. The axon hillock is the location that we will determine whether or not we send a message, right? So this is the determining spot. If we, and we'll get into it, reach threshold at this spot, we'll send a message. In this situation, we can see, I've mentioned something called myelin. You can see that there's this pink coat on the neuron. And that is produced by those Schwann cells, assuming it's in the PNS, and that will help that message jump. As the message gets to the end, we know that we will again release some chemicals, from the axon terminals. And that's how neurons work. Main goal, receive stimulus, decide whether or not it's going to send a message, send said message. Once it starts, it happens automatically, and we'll talk about the intricacies of that. And then when it gets to the end, it releases a neurotransmitter, and guess what? Most likely, there's probably another neuron over here that's going to decide whether or not it sends a message, right? So we can see the cell body, the dendrites, the axons, the axon terminal. Obviously, in here, we'd have, let's probably bad color, we'd have some patches of Niesel bodies found in that cell body. Right? Input, output, straightforward. This shows the same exact thing, but it's showing you these action potential propagations via the arrows in that unidirectional flow. You can only send a message in one direction. You can't send messages in a neuron forwards and backwards. It always goes in the same direction, assuming it depolarizes, and that breaks down kind of the roles of some of those structures. This is showing, and we'll come back to this picture when I talk about neurotransmitters, but the only thing this is showing, look, this is a presynaptic neuron talking to this guy. He makes the decision at the axon hillock. He sends a message, and this is highlighting something called a synapse. So whenever you have a neuron talking to another neuron, the junction where both of those neurons are meeting is called the synapse. And at this synapse is where the magic happens, right? That's where the neurotransmitters, which are these chemicals in here, that's where the neurotransmitters are released, right? They will bind to said receptors, and then that will cause the electrical message to basically transfer from one neuron to another. So synapse is a key term, and neurotransmitter is a key term, right? So let's say this is just showing you here a sensory receptor, right? In our skin, and we'll talk about a variety of different types of sensory receptors, but in our skin, we have these receptors. The second that needle in this picture goes through the skin, what it's going to do is it's going to trigger one of these pain receptors. And then what do? Once that gets triggered, we'll talk about how, it's going to send a message via an action potential, synapsing with another neuron, delivering that message to the brain so we can feel the sensation of pain. Now, in order for a neuron to send the message, right? This is kind of showing you a general overview, and then we'll talk about actually what's happening in the membrane of the neuron. So if you have a neuron, draw it like this, right? It's got these things at the end. It's not the best neuron picture. Whatever. That's the cell body. And let's say we poke this neuron. Maybe it's some touch or pressure or something like that. Maybe the charge of the neuron changes a little bit. Nothing happens. Maybe we push a little bit harder, right? You still wouldn't feel it. Maybe the stimulus is stronger. And then maybe you put enough pressure on it that you can feel it. So basically what happens, and we'll talk about this in a little bit in some more detail, strength of the stimulus, right? Let's say there was 3 stimuli. This guy would probably send a message. If there was only 1 stimulus, maybe it goes, and there would be no message sent, right? So the key factor that we're introducing here is that all neurons have what we call a resting membrane potential. And because I have my picture of my neuron here, I will show you. The resting membrane potential of a neuron is slightly negative on the inside and slightly positive on the outside. The significance of that is when you look at this chart. And what is this showing you? This is showing you the charge of the neuron, and then this is showing you time. The key thing here is that black line. This is the neuron's resting membrane potential. This is when it's not doing anything and it's just chilling and it's not sending a message. It starts off negative. Specifically, which knows it starts off at negative 70 millivolts, negative 70 MVs. As this neuron gets stimulated, the charge goes up a little bit. A little bit of positive ions come in. As the neuron gets stimulated more positive ions come in, and that charge gets higher. A message will not be sent unless this charge reaches what we call threshold, right? And we'll dive into it. Threshold is at negative 55 millivolts. So the key thing here is you could tickle a neuron as much as you want, but unless the charge reaches threshold, it does not send a message. Once the message is sent, you can't go back, right? So once it reaches threshold, neuron depolarizes, and that message will be sent all the way through, clearly releasing neurotransmitters at the end. So the way neurons work, they either send a message or they don't send a message. A couple key factors: resting membrane potential, and threshold, which was at negative 55. Why? There's negative stuff inside the neuron, inside that axon, positive stuff on the outside, right? So this is showing you, if you were to measure the charge, right? Slightly negative on the inside, slightly positive on the outside. And what does this show? This is just showing you the plasma membrane, this part, that red line. This is showing you a zoomed in version of that plasma membrane. And we can see that the resting membrane potential is at negative 70. Why is it at negative 70? The inside's negative. We know that. What makes it negative? A few things, and these are key factors. One of the first things is we have leaky channels called potassium. Potassium's the K plus, and sodium. Sodium is the Na plus. So there are these leaky channels that are constantly allowing these ions. Basically, all of the electricity is controlled by ions. Some ions are positively charged and they move in, makes the cell more positive. Some ions are negatively charged, they're inside, makes the cell more negative. So we have a couple key factors here. Number one, we will see that there is a high concentration, find it good, of sodium ions on the outside. And again, sodium is the Na plus, right? A lot of salt on the outside and a lot of, I'll write that here for you, Na plus. And the potassium, the K plus, a lot of potassium on the inside. They're both positively charged ions, but they are, because there's the leaky channels, right? They are usually, moving back and forth, but in higher concentrations, salt on the outside, potassium on the inside. And I will show you specifically how those concentration gradients are formed. But the negativity inside the cell is because of more structures. We have lots of organelles in there. We have lots of proteins, right? You can see the presence of this protein. These proteins are negative, and that's significant. And lastly, we have another structure, and that is this big guy right here. This is important. This is called a sodium-potassium pump. The protein, the enzyme itself is called sodium potassium ATPase. And the reason why, as you can see here, is it uses energy, aka ATP, to pump sodium ions out and pump potassium ions in. This is why you have a high concentration of salt outside and a high concentration of potassium inside, because this pump is actively using energy to force these against the gradient and maintain that negative resting membrane potential. So key things: salt outside, potassium inside, negative things inside like proteins. And how do we maintain that resting membrane potential, or the fact that salt's high on the outside and potassium's high on the inside? Via the ATP using sodium potassium pump, right? Key thing: ratio 3 sodiums to 2 potassiums, right? ATPase, more potassium, cool, right? So that is explaining the cellular component of the membrane, what's where, and kind of how we establish that resting membrane potential and how we establish those ions in the correct space. So we have two types of behavior that can happen at a neuron membrane, right? The first thing is that the membrane potential, which we know is at -70, it can get lower. If it gets lower, that means it's more negative. How does something get more negative? By losing positives. It's that simple. Potassium is positive, and we know it's high on the inside, right? So if we had a channel that would open up, aka like a voltage-gated potassium channel, which is important to know, it opens in response to a charge. If we lost a positive, the inside of that would be more negative. So we call this a graded potential, and I'll explain to you the difference of what that is versus an action potential. And that is called hyperpolarization. Hyperpolarization is when the cell gets slightly more negative. And we can see that right here. Depolarization, which is a term I've already used in this lecture and will continue to use literally for a very long time. Depolarization is when a cell gets more positive. So you're at -70, and now some positives are coming in, making the inside of the cell more positive, and that is caused by the influx of sodium. So when potassium leaves, the cell gets more negative. We call that hyperpolarization. When a cell gets more positive, we call that depolarization. And if you recall, again, we'd have to reach that magical threshold line. And depolarization is caused by positive things coming in, sodium, and that inside of that cell gets more positive. So two key words, very, very important to recognize that. And how can we depolarize a cell? There's a couple different things that are going on. In general, we have a few types of membrane proteins. This is showing you one, and it's explaining how it forms a graded potential. I'm going to use this example, and then I'll show you, let me see here, 4 types of channels, but then I'll give you some examples. So I'm going to use this space to kind of explain this. If you have a neuron that looks like so, and we have our graph with a resting membrane potential, that would be, -70. If I were to poke this thing and make it depolarize, it would go bloop, and nothing happened. So a small localized change in membrane potential is called a graded potential. There are multiple ways to generate this graded potential. Let's say I poke them twice. Boom, 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 boom. What happened? That looks like 3 times, but it doesn't matter. 3 times. Nothing. Why did nothing happen? Yeah, you got a little positive for a second. Nothing happened because it did not reach threshold. Let's say I poke this guy or put a lot of pressure or poke a pin through the finger and it hits these sensory receptors. Graded potential, boom. Once there's enough graded potentials and we reach threshold, the message is sent. So, what things can cause graded potentials? Obviously, once we reach threshold, right? We send a message, we're going to squirt out some neurotransmitters. That's how it works. So some things, try to clean this up for you a little bit here. Some things that can cause graded potentials are channels. The first one we have is a mechanically gated channel. Mechanically graded channels are found in receptors in the skin for pressure and things like that, but it opens in response to a mechanical stimulus. So when I sit here and I touch gently on my finger and I don't feel anything, right? There's not enough stimulus to open up enough channels to have me feel it. Once I put pressure on it, those channels will physically open up. What happens? Positives come in, reach threshold, depolarize, send a message. So a mechanically gated channel opens in response to pressure. A ligand gated channel, and again, these are just different channels that allow for graded potentials to depolarize a neuron. A ligand gated channel opens in response to usually a chemical, aka a ligand. So some of those neurotransmitters that we've been talking about that get released can bind to a neuron, aka bind to a ligand gated channel, and it will cause depolarization. Right? If that chemical binds, it opens up a channel, positives come in. Mechanically gated, if I put pressure on something, the channel opens, stuff can come in. Ligand gated, chemical binds, opens up, stuff can come in. And you have, let's see. Cool. Not only can you make positives come in, but you can also allow for negatives to come in. So the difference here would be a ligand-gated channel that allows for hyperpolarization. And I'll show you this chart real quick because we always come back to the chart, right? If it's here, a depolarizing stimulus would go up, a hyperpolarizing stimulus would go down. So in this case, you could see that this channel specifically opens up and it allows more negatives to come in. And what would that do? Make the cell more negative and thus a hyperpolarizing graded potential. Simply put, graded potentials are small blips in the change of the membrane. They could be positive or they could be negative depending on the channel being opened. We talked about, let's see, leaky channels. Oh gosh. Leaky channels, right? The potassium that's constantly moving back and forth, the sodium that's constantly moving back and forth. So these are called leak channels. We talked about ligand-gated channels. We talked about mechanically gated channels. And now there's a fourth one. And the fourth one is these are all extraordinarily important, but the fourth one is particularly important. And this is where we see a channel called a voltage gated ion channel. And what a voltage-gated ion channel does is pretty much exactly what it sounds like, is it opens not all the time like leakies, not because of a chemical, not because of some mechanical stimulus. It opens when the membrane potential changes. Simply put, if you have enough depolarization, that thing opens up. This is extremely important, and I'll go back to our neuron picture. They're getting worse, but that's okay. Because all of those graded potentials so far I talked about over here, over here, over here. But once that message gets started, these voltage-gated ion channels are what open up along the length of that axon. As the charge changed, channel opens, positives come in, opens the next channel, positives come in, open the next channel, positives come in, and we'll see a picture in a little bit. But that's where those voltage-gated ion channels come into play, is allowing that action potential to move along the length of the axon. Major difference, graded potentials versus action potentials. Action potentials, the entire thing that gets sent. Graded potentials are small changes in the blip of the membrane potential, usually near the cell body. This summarizes the 4 types of channels, gives you a description and a location of where they're usually found. I talked about it on the neuron. So if you have access to this, check it out, be familiar with it, where we find them and what they do. Here, we're looking at kind of, and I've drawn you the picture 100 times, the different anatomical structures of a neuron, from the dendrites to the cell body to the axon. And here we see that axon hillock, which is extremely important. That is the part where that decision process takes place, if you will. And we can also see the axon terminals. And it tells you what the functions of these are, right? We can also see that we have color-coded locations of the variety types of channels that we've talked about, whether they be chemically gated channels, right? Ligand stimulus, voltage-gated ion channels, etc. So here we see that trigger zone, and we see 2 terms, EPSPs and IPSPs. I actually told you what those were already, but I didn't use that term, and when we get there, I will explain it. So this is the main guy here that I have highlighted in blue. The other 3, right? The main guy is called the postsynaptic. So we talked about what a synapse is. A synapse is where other neurons meet. Here we have 3 presynaptic. If this is my postsynaptic neuron, this is my presynaptic neuron. What are they going to do? They're going to talk to each other. How do they talk to each other? Various chemicals, right? So I don't know if that picture helps you or not. We have a scenario where 3 different neurons are trying to send this guy a message. That poor guy's got to decide. All he can do is either send a message or not send a message, right? So these 3 guys are sending messages. Some of them can be, as we discussed, exciting the neuron. That would be an EPSP, excitatory postsynaptic potential. That excites this guy, and then big picture, what are we looking at? Right? If he's getting excited, what's going to happen? He's going to get more positive. Or maybe some of these guys are sending a IPSP. They're telling this guy, yo, don't do anything. And maybe they're making him more negative, right? That would bring this membrane potential down. So what we have is basically a battle going on, if we recall, threshold, right? Negative 55 millivolts. A battle of consistent fluctuations of positives and negatives. And the question is, does it reach threshold or does it not reach threshold? So these neurons all talking to this guy are sending positive messages, negative messages via chemicals. And here at this axon hillock will be the decision-making process of, do we send an action potential or do we not? So simply put, graded potentials will either help it reach threshold and thus send an action potential, or they will not. And that is what EPSPs are and IPSPs. And that is the relationship between presynaptic neurons and postsynaptic neurons. Clearly, the one that's closest to the axon hillock will have the most influence, right? The other guys that are kind of far away, not as much. But if there's enough of them, they can have some significant impact. So hopefully that makes a little bit of sense. We keep referencing that chart, right, with the charges, because that's kind of an important thing to have in your head visually. And here we specifically introduce, we already did, your excitatory postsynaptic potential. Makes the inside of the cell more positive, aka it will depolarize, right? Making it easier to reach threshold, right? So if we look at the picture, your EPSPs make it go up. Your IPSPs make it go down. Makes sense. Inhibitory postsynaptic potential make the neuron more negative. That's why the charge goes down. And they make it harder to reach threshold. And this is our trigger zone. That's the axon hillock that we've talked about. And key thing, if that region, we don't care what happens over here. But if that trigger zone reaches threshold, we send an action potential. That's how it works, right? And in reality, we got a lot of messages being sent to this neuron, and that it makes it easy for him to make a decision. Does the axon hillock reach threshold or does it not? And who cares how many messages it's receiving? It is simple as far as, is threshold met or is threshold not met, right? Because we have this constant bombardment of excitatory and inhibitory messages. Does he send a message? Does he not? And all of that is happening at these synapses. Certain chemicals are being released that either excite or inhibit that postsynaptic neuron. I kind of talked about this already, but it just shows you another view. Maybe this is a picture I drew you a couple times. As far as, pick a good color, signals coming in to the dendrites, right? We have the obviously positive ions are going to be in there. And once we get to threshold at that axon hillock, the action potential, and here we see the term action potential, will literally propagate along the length of that axon to the ends. And then you know what's going to happen here. Obviously, some chemicals are going to be released. Maybe onto another neuron. Probably. So that's great. So you have a nice picture that represents accumulation of graded potentials and the difference between graded versus action potential. This is showing you, let's see, threshold. How do we get to threshold? Right? In this situation, we've reached, right? And it's showing you how the kind of, I guess it doesn't really show you the channels, but as we open up, right, we reach threshold. This is our axon hillock. We have channels we've discussed called voltage-gated sodium channels. Once the threshold is met, and we talked about this, it's just showing you in a slightly different picture, those channels open up because they open up in response to a change in voltage. Why do we have a change in voltage? We met threshold. Once threshold's met, sodium comes in. What does it do? Make the cell more positive, right? It was negative, but guess what's going to happen right here, right? This next channel is going to open up. Sodium comes in. Guess what? Next channel opens up. Sodium comes in. Next channel opens up. Next channel opens up. And what we have is kind of this inching of positive charges moving down through the neuron. That's our message. And this shows everything we talked about on one picture, a very important picture. We have the chart that shows an action potential. This is what it looks like, right? These are stimulus. These are obviously EPSPs or graded potentials. We didn't reach threshold. Nothing happens. Once you have that significant enough stimulus here, the message is sent. It will repolarize, get back to normal, so it can send another message. So the keywords that we have here, and I'll try to color code this for you, are depolarization. It will always jump up to about positive 40. And that is because of sodium ions coming in. Depolarization, depolarizes, salt comes in, cell gets more positive. That's why the line's going up. Once it does that, we have a natural efflux of potassium ions. So think, so positives are coming in, thing goes up. Positives go out, cell gets more negative. That's what brings us back to our resting membrane potential. But it happens so rapidly that we have a small period of hyperpolarization, and then we will bring it back to the resting state via that sodium-potassium pump. Right? It pushes the salt back where it's got to go, pushes the potassium back where it's got to go from a 3-to-2 ratio, bringing that back up to our resting state. And what can we do next? Send another message. And that's the chart. And you should totally know it. This tells you exactly what's going on as far as the channels, right? So we reach threshold here, right? Voltage gated sodium ions channels open, right? Start to close. Voltage gated potassium channels open. Then they start to close. And that is why these ions are moving back and forth, creating the electrical message. And this is just showing you the same thing. It's showing you what channels are involved in each state, whether it be resting, depolarization, or hyperpolarization. Right? Be familiar with it. Obviously, those leaky potassium channels are always open. So just always floating back and forth. And then we talked about the propagation through the axon. Right? So it's really just one general concept of how do we depolarize and send a message through a neuron that has a million pictures to it. And you got to kind of understand the intricacies as we start going through a variety of other topics. It's all going to come back to this, right? You have a lovely chart here. Tells you how long it takes, milliseconds. Cool. But once we kind of get this depolarization happening at the trigger zone, we have this chain reaction, right? Salt comes in, cell gets more positive. What does that do? Open up the next channels. What happens? Stuff comes in, gets more positive. What does that do? Open up the next channels. Sodium comes in, boom, gets more. So you can see that it's inching its way down, right? As it's inching its way down, the potassium channels are opening, allowing that positive to leave. As that positive leaves, right? What was my other color? Red. As we depolarize, trying to color code it for you. What does that do? Open up the next channels. Boom. Potassium leaves. And we kind of depolarize and repolarize. So I like to visualize this um, as kind of this wave moving through the neuron, right? It's going to move through the neuron. I'm not going to really be able to draw this as an action shot. Then that wave moves through the neuron, moves through the neuron. And what did it do? It just inched its way down till it got to the end. Chain reaction. Once it starts, automatically sent. And that is an action potential. And we have two different ways we can send this. We can send them fast or we can send them slow. The only difference, we have saltatory conduction and we have continuous conduction, right? Obviously, some neurons in the body want to send messages faster. They will use saltatory conduction. If you have to send messages faster, you will have myelin. I'll try to color code this for you here. You can see the presence of myelin, remember, made by Schwann cells or oligodendrocytes, depending on if it's the PNS or the CNS. You have the presence of this myelin creating gaps. These gaps, gap, 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 are called nodes of Ranvier, right? They're areas of unmyelinated neuron. And what happens here is the action potential, which we just talked about forever, can jump from node to node. So just thinking about it, there's less walking, right? That whole membrane has to depolarize, repolarize, depolarize. This is like depolarize, repolarize, depolarize, right? I like to relate it to walking, which you would see here. Got to do this as I erase it. Got to do this along the whole thing. One foot in front of the other, very, very slow, right? That's continuous conduction because that depolarization is occurring across the entire membrane. But here, you're running. You take a step, go forward 3 feet, whatever, 4 feet, depending on your gait, take another step, take another step. So we get a faster transmission of that message because it gets to jump those gaps. Why? Myelin. So what does myelin do? Speeds up an action potential. Myelinated axons will send a message way faster than an unmyelinated axon. And we see that node of Ranvier, and that is the gap in between the myelin sheaths. Got it all there. Continuous versus saltatory conduction. Be familiar with both of these terms? You're good. How do we make myelin? Well, we know, because we talked about it, that Schwann cells and oligodendrocytes make myelin. The difference is, and this shows you how it happens, right? You can see that Schwann cell surrounds it, starts producing it, and we have this myelin sheath here. Schwann cells are found in the peripheral nervous system. They're going to myelinate small sections of an axon. Oligodendrocytes are found in the CNS, and they can usually myelinate multiple axons. They have a bunch of arms that come out and can add some myelin. So we talked about that. PNS, right? We're going to usually have one axon. And we can see those nodes of Ranvier. We can see the Schwann cell. We can see the myelin itself, right? PNS. Talked about the node of Ranvier. Awesome. Just another picture if it helps you visualize that. These are the areas where the depolarization is taking place. Makes it faster. Saltatory conduction. Showing you another picture, right? What happens? Depolarization at the nodes only, right? So it makes it faster. Less ions need to move around. Genius. The oligos myelinate in the CNS, right? They can myelinate several axons. You can see that oligo here. He's reaching out. He's throwing a little bit of myelin there, a little bit of myelin there. Oh, this is a different guy. He's throwing a little bit of myelin there. So they're, very condensed. Obviously, the CNS is going to have, way a lot of neurons in there. So it kind of suits his purpose perfectly that he can myelinate multiple axons. So we have the concept of neurotransmitters that we've talked about. Those are the chemicals that are released, right, from the synaptic knobs, and they go in. Some of these neurotransmitters can be excitatory, and they'll make this cell more positive. Some of these neurotransmitters can be inhibitory, and they will make that cell more negative, respectively, right? Ligand-gated channels. When a thing binds, it either makes it positive or it makes it more negative. So these are some examples. Acetylcholine is something we talk about a lot, right? ACH, we're going to actually talk about it a lot. Neuropeptides, I'm going to show you a few of them and kind of tell you what their role is and where they take place. Nitric oxide and a variety of other things as well. Neuropeptides are not necessarily, well, if you look on here, it is a category of neurotransmitter, but they are kind of their own division. And we have some examples. So substance P is one, endorphins are one, and a lot of people have heard of this. Angiotensin, CCK, these deal more with blood pressure regulation and digestion in the case of CCK, right, which we'll talk about in the future. So I want you to be familiar with a few of them. Substance P is involved in pain, right? Enhances pain perception. And obviously, you have different types of neurons. Some of them release substance P, some of them will release ACH, some of them will release whatever. That's their job, pretty much. Endorphins and things like that deal with inhibiting the pain, blocking the release of substance P. So an endorphin, like if you're talking about your runner's high and stuff like that, even if your legs are hurting, you usually don't feel it. Things of that nature. Once we release, this is one of the last concepts as far as neurotransmitters go, right? You got your neuron, sends a message. Talked about that forever. Chemicals are released. Now you can't just have, right? Let's say this is our other neuron over here. You can't just have these chemicals living in that synapse forever. We have to get rid of them, right? So there's three ways that we get rid of them. One of them, they just diffuse away. They get released, they diffuse away. If they're no longer there from a high to low concentration, they can't keep binding to this thing, triggering him, making him send a message. Another way is we have enzymes that are in here that I'm going to draw like this Pac-Man looking thing that just chomp it up. They live there. Chemicals get released, they do their thing, enzymes come around and break them up. That's called enzymatic degradation. And the third way is they get taken back, which is a very efficient way. You release them, and then the cell can take them back in to reutilize them. So know this, there are three different ways that we can get rid of neurotransmitters at a synapse or at the synaptic cleft. And we're going to kind of come to the end here as far as structural classifications of neurons, right? They can be classified based off of a number of different things. Here we're talking about the number of processes. So how much stuff is coming off the cell body, right? Specifically the dendritic processes. So we have three general categories. We have a multipolar neuron, and you can see there is a lot of stuff coming off that cell body. What cells, star means important, What cells are multipolar? Motor neurons. All motor neurons and some other stuff that we find in the CNS are multipolar. We have a bipolar neuron. How much stuff is coming off? Two branches. Bipolar. Makes sense. Where do we find these? Sensory neurons, usually special senses. Eye, ear, nose, things like that. Don't usually find them elsewhere in the body, specifically related to special senses. And then we have a unipolar, or they call a pseudo-unipolar, and there's only one branch coming off. Unipolar. Sensory neurons in the PNS, usually when we're talking about the skin, things like that. Sensory neurons are unipolar. So we kind of think of multipolar, motor, unipolar, sensory. There are a few rare exceptions, which we'll talk about when we get there. We have Purkinje cells and pyramidal cells. When we talk about the brain, we'll go into some more detail. But Purkinjes in general, you can see they have a lot of dendritic branches coming off, but not directly off the cell body. There's that split, and then we have tons of dendrites out here that could receive a boatload of information. And those are found in the cerebellum, part of the brain. And our pyramidals are found in the cerebral cortex. So clearly, when we get there, we'll talk about that. That's brain stuff. And lastly, and this kind of prefaces for our next lecture coming up, we have, let me see if I could do this the right color, motor neurons, sensory neurons, and interneurons. Important. Our sensory neurons, right? We could see this guy here. That's our unipolar neuron. It's going to receive some stimulus, transmit this message in. The cell bodies are found in a structure called the dorsal root ganglion. In lab, we'll talk about this. For lecture, you don't have to stress about it too much yet. And it sends a message into the spinal cord. Sensories send message in. Sensory sends messages to the CNS. Then we have some type of integration. So this is our interneuron. I'll try to do this a little less confusing for you here. Boom. And a variety of things that we'll talk about with that. And then we have our motor neuron, right? Don't forget talking, they're sending a message. And then this motor neuron will come send a message out to some type of muscle or gland or some type of effector. The key thing here is we can see this multipolar neuron. We can see the unipolar sensory. We can see that sensory comes into the CNS. We can see that there's some integration, and we can see that motor comes out of the CNS. This specifically is showing you the spinal cord, which we'll talk about in the next lecture. And this kind of wraps it all together, right? If you have a scenario of a reflex, which we'll talk about, where you have this pin going through, these are our receptors, right? Mechanically, right? Gated channels that are going to open up in response to some stimulus, whether it be pressure or whatever. If you have enough EPSPs, then we send an action potential. You have to reach threshold there. That action potential moves along the axon via that process chain reaction we talked about, where sodium's coming in, potassium's leaving, hyperpolarization of the sodium-potassium pump. Then we have integration here, right? Once the action potential reaches the end, there's neurotransmitters that are going to be released. And then this will cause an EPSP, right? Again, if there's enough stimulus, then that message will be sent. Same thing happens here. If there's enough stimulus, we will send it out, and then we will result in contraction of our muscle, which might cause, right? Obviously, if somebody stuck you with a pin, probably pull your hand back, happens super quick. All these messages are being sent very, very rapidly, especially through myelinated neurons. Makes sense. This is kind of putting it all together. And this shows you the same thing, right? Just without all the cartoony pictures. Um, and you can see here that we have the presence of usually unipolar, usually multipolar. The action potential is transmitted. Neurotransmitters are released here. Right? That's the big picture. That's how the nervous system works. It's just a series of neurons that are communicating with one another through various circuits, and all the movement of ions basically happens by itself. We don't have to control that, right? It's regulated by chemical influence. Um, and this kind of prefaces for next lecture as well. We're going to talk about nerves and the parts of a nerve, things like that. I will mention it briefly. A nerve is set up like a muscle, right? We know a nerve is a group of, we started off with that today, a group of neurons. So all you do is you take a bunch of little neurons. You can see this one has myelination, right? Little nodes of Ranvier. You group them together, right? And then that has a connective tissue layer on it. Then you group these together, then that has a connective tissue layer on it. And basically, it's a way to organize and kind of keep everything where it's supposed to be. And the connective tissue layers that I'm discussing are called the epineurium on the outermost portion, the perineurium, which is surrounds the fascicle, and the endoneurium, which will surround our individual neurons. It's a way to organize everything. Key here, a nerve is a group of neurons. As we get into spinal nerves, we will talk more about that. This is a picture of it. We'll wrap it up with a couple disorders. So we have MS, or multiple sclerosis, right? Is an autoimmune disease that causes progressively destroys or progressive destruction of the myelin sheath, right? So here, that myelin sheath is being destroyed. Not 100% sure about why, right? Most autoimmune diseases aren't really fully understood. But clearly that's going to affect the ability for this destroyed neuron to relay messages. And it could lead to muscular weakness, vision issues, abnormal sensation, right? There's a variety of type of neurons that are myelinated. That's MS. And different types of neural disorders in the brain. Talk a little bit about this when I get to the brain itself. But there's several types of depression, major depression. You can see here you have manic depressive illness associated with bipolar depression, seasonal affective disorder, right? Common feelings of lack of interest, sadness, right? A whole bunch of other stuff. And they are treated by SSRIs. So these are selective serotonin reuptake inhibitors, and they work on that neurotransmitter, that synapse there, that affect the reuptake of serotonin, which is a neurotransmitter, and are used to treat some of these conditions, right? Epilepsy, and we have various types of excitotoxicity. Yeah, I mean, you could read it. I might ask you one question about it or something like that, but I'm not going through the specific physiology. And that wraps it up. So what did we talk about? The main thing is be familiar with the terms, everything I highlighted there. Obviously, I'll post like a little study guide to kind of help you tailor and focus some of the what we discussed. But it's once you get the hang of it, it's not too bad. What is a neuron? How does it work? What are the parts? How do we send messages? How do neurons communicate with one another? Were all topics that we discussed here. And then we kind of finished it up by talking about characteristics of sensory and motor, which we will obviously progress through in the next lecture. So I will see you there.